All right, psychology nerds, welcome to another episode of Psychology and Stuff, the podcast from Good Cause Productions at the University of Wisconsin, Green Bay. I'm your host, Allison Jane Martingano, and today I'm joined by Dr. Jason Cowell and Dr. Todd Hillhouse, talking about the brains of both humans and rodents. Both are psychology professors at UW Green Bay. Dr. Cowell studies the neural development of moral judgment, empathy, self-control, and pro-social behavior in childhood, adolescence, and adulthood. Dr. Hillhouse studies neuropharmacology, especially addiction and pain. His lab uses animal models to better understand and develop treatment options for these disorders.
I'm Jason Cowell. I run I guess it's a changing name, but Developmental Social Neuroscience Lab on campus. We do a lot of human research where we're kind of two different programs. On the one side, we're doing a lot of collaborative work where we are testing kids' brains as they start to learn right and wrong, caring for others, empathy, different things along those lines. We're trying to see how fast and how much they process those things using EEG. In order to train for that, we've also been doing a lot of adult studies, and all of those are more on more complex self-regulation, perspective taking, and morality as it relates to reporting bad things that happen when you were younger. So early life stressors is what we call them, but things like divorce of your parents, things like parental mental health when you were growing up, and how that kind of shifts how efficiently your brain can process different things by adulthood. So yeah, I run a EEG lab on campus with about 10 undergrads a semester that we are actively testing kids and adults.
All right, and for those that don't know, EEG, that's the one where you stick it over your hair, right?
It is. It's a cap with a bunch of little electrodes that all slide on your head, and they theoretically measure electrical signals that are being generated from within the head.
All right. So you're not actually sticking anything inside people's brains.
We are not, but there are versions that do that.
Oh, okay. All right. Well, we might get back to that later.
We'll come back to that, yeah.
All right, Todd, how about you? What do your lab do?
I run a behavioral neuroscience lab. We use mice as our primary subjects. And so the overall goal is to develop new medications for depression, pain, and addiction. There's a big overlap between all three of those areas. So we are looking at kind of the same receptors for those different disorders. Another part that we're really interested in is developing better models of translation. So we understand that, a mouse is a mouse and a human is a human. So we're trying to make sure that the assays we use or the tests that we use are as close to what we see in humans as possible. And then we recently started a second line of research with a collaborator on campus here to develop, it's more human-based, it's to develop an app to measure like drug intoxication. Okay. So we can have rapid testing and seeing if people are intoxicated in a workplace.
So I'm curious, how do you test mice? Because obviously you can't, ask them questions, and I'm assuming you can't put a little neuro hat on their heads. So what do you test for them?
So we try to design different assays or tests. So for example, in our lab, when we do our pain assays, we can use models that mimic humans. So we use what are called pain-depressed models. So what we do is we give them a mild pain state, and what we see is we see a reduction in behavior they normally would do, like moving around, just walking, or pressing a bar for a food reinforcer. And what we see is that, we can get them to respond at a stable rate. If we introduce some type of pain state, we'll see a reduction in the behavior. And if we give them something like ibuprofen, or something like morphine, we can actually see that come back to normal, like we would see in a human person.
So you're watching for the behavior of the mice.
Yep. Our lab primarily looks at behavioral outcomes.
Awesome. And so do you videotape them to do that, or do you watch them with your eyes?
All of the above.
Okay.
So we use a combination of, we have video cameras, we have tracking that can score it. Sometimes we score videos by hand because it's too difficult for the tracking to pick it up. And then we also have automated. We have quite a few pieces of equipment that are automated. So they just do it all for us. We put them in, we hit go, and then this spits out the data. Those are the fun ones.
Yeah, wow. So it's amazing. It's amazing that I've worked with you for as long as I have and don't know the specifics of what you do in your lab. I wonder if you could talk a little bit, and let's go back to you, Jason, on the biggest challenges and practicalities in running your lab, and then maybe some of the biggest pros.
Yeah, and Todd and I have talked about this a lot. The challenges are different, and I know Todd will comment on his lab too. Not to say I don't have to keep my subjects alive, but it's not my responsibility to feed them, to house them, to everything. And so there's a big benefit to human research that you don't have to take care of all of the daily necessities for them. The disadvantage comes, you then have to work around when they're available, if they'll come into the lab, when they can be tested, when they don't show up. So running a study, while you don't have to keep them housed, et cetera, you then have to work around their schedules. So this is especially present in kids studies where honestly costs to run kids have risen exponentially because you're asking families to come in on spare time. So after school hours, on the weekends, you're trying to get undergraduate researchers as well, several at a time that can show up at the same time, can do this. And honestly, most of the grants that we've been writing, we're paying families $30 to $50 to come in, and kids get toys and all of this because we have to value that time as well.
Right.
And yet we're still having 50% no-show rates, meaning just people have so much else going on in their lives and you're so dependent upon that. And so I think that's one of the issues is on finding adequate sample sizes. So in an EEG study, I love to have at least 100 people in my study just to really make sure that that the findings I'm finding aren't just because I have a really small sample and it's not a true thing. It's just because of the small sample. To do that can take me 4 or 5 semesters a lot of times to get to that 100, just because even if we schedule 50 to 60 a semester, at a 50% no-show rate, that's 25 a semester. And so you're slowly building this over time. I'd say that's the biggest challenge. The exciting part is that it translates really well because you're dealing with humans. So every behavior, if I want to know how empathic someone is, I can ask them. I can do, as we've talked about, AJ, substandard empathy tasks with them. I can actually test them on a series of things that have a direct translation to what you expect in the real world. So, and I think that's a big challenge that you'll end up talking about shortly. So that's the awesome side. On the kid side, the largest issue is that the questions you want to ask, practically speaking, you can't ask with kids. So the biggest questions that always come in neurodevelopmental are, do the structures or the structural connections change across childhood? So I think everyone wants to know, hey, when does full developmental prefrontal cortex happen? To what extent is efficient prefrontal, so the area in the front of the brain that's primarily responsible for self-control, working memory, different tasks like that we know develops from about age 3.5 to age 26-ish, and we know that the front end of it is the development of a lot more connections happening there up till about age 8. And then after 8, you start to get the pruning of certain things and more efficiency and fatty tissues around that allow it to translate signals more effectively.
What I'm learning from this is that my brain gets fatty at the front. And
it gets all fatty.
Okay,
No,
that's cool.
The axons get fat that allows them to have a better electrical signal that is transmitted. And I think the coolest questions can't be answered because in order to do that, you have to put a kid into an fMRI or into a magnetic resonance imaging machine. And the problem with MRI is that if a kid moves more than a couple of centimeters, the picture that you get is blurry. And so speaking, most of my graduate school was actually doing fMRI with 6 to 12-year-olds. And we tried a couple of 5-year-olds and a couple of 4-year-olds. And we lost 3/4 of our data every time because you just couldn't get the pictures right.
Yeah, how'd you get a 4-year-old to sit still for 10 minutes?
Don't. That's the problem. So there are different functions to try to do this where you have padding that goes around their head to try to remind them how they can't move their head. You try to have games that their hands are slid into so that they can fidget without it affecting any kind of movement, but all those cool questions are gone. So anything where you see a pretty picture of a brain with a splotch over the top that you say, oh, this is the place that's activated, you really can't do with kids. And those are the questions you actually want to ask. You have to use other technologies like FNIRS that only work if there's no hair on that part of the head and only work a couple of centimeters down into the cortex. So you're not getting the full picture because of the practical sense.
All right. So what I'm hearing is that research with adults is much easier than research with kids.
In so many ways.
Oh yeah, and that's the good. Never work with animals and kids, right? That's the thing. You guys are doing both. So Todd, tell me, how's it different with mice?
So the exact opposite of what we run into. So the big issue for us, I would say, is cost of animals. They just continued to rise. And then keeping them alive, right? So we are responsible for them, and we have to check them every single day. That includes the weekends, that includes over Christmas break or all the holiday breaks, Thanksgiving break. And so that can be really difficult, especially with kind of the changing economic times and the changing student population. Most students work full-time while they go to school full-time, and so it is hard for them to give up time. So I would say that is probably the most difficult thing. Is it's very stressful. For example, I got a call on Sunday saying that the lab was significantly cooler than it was supposed to be. Mm-hmm. And so we had to rally the troops and contact a bunch of people, and luckily we were able to get the room up without having to come to campus. But those are the types of things where I've had to leave family events to come and check on mice or to help out in that way. So that is kind of a downfall. On the flip side of it, though, is that it's much faster than human research, depending on how you have your lab set up. I don't have to recruit. I purchase my mice, or we also breed mice. So they are there when we are there. And so it allows us to be able to ask questions. It allows us to be able to do research any day that we would like to do research. So I do like that part of it. That helps me produce quite a bit. So, my goal usually is to try to get a publishable unit each semester, or at least one per year. So I try to design ahead of time. I usually have about 6 to 8 students in the lab, so we have multiple projects going at a time. So we're not fixed on just doing one project. I usually have 2 different projects, or I have people running data on multiple projects. So again, it just allows for a lot of flexibility, a lot more control. I have done some human research, and it's been very hard. One of my studies, the one human study I have published, I had students come back 4 different times. We ended up with an N of 8. I think we had like 20, and we just couldn't get them all in. So I do appreciate, the benefits we have, but the cost is also expensive because we have to buy syringes and gloves and just the daily lab supplies, food and bedding. Yeah. So it just adds up in terms of cost-wise. But I did come from poor labs, so I have learned how to become economic is how I like to pitch it. So we actually do produce a lot for the very small budget that we have. It's just about being efficient and kind of utilizing all that we have.
Yeah, and I should say for both of you, you're not doing research with children and mice, but you both are training undergraduates all the time. Yep. And that obviously is one of the great strengths of the research that you do here, is that undergraduates are learning, but that also means you're constantly training new research assistants every semester, I'm guessing. Yeah.
Yeah, I think yesterday I was in my lab and over a 2-hour period, 3-hour period of time. I think from 8:30 in the morning, I got to the lab with my students. I taught at 11, and between that time period, I went back and forth from my office to my lab training students, but also trying to get my work done about 7 times yesterday.
Is that a long distance you got?
We're going from Mac Hall to lab science. Yeah, so if you take the tunnels, it's a little further, but I got my steps in.
Okay, good. Well, and I'm glad we got the tunnels, so you didn't have to go outside as it's starting to get a little cooler out. All right, I'm gonna move into what I'm going to call the ethics section. So I'm going to make some broad assumptions here that you can do a lot more to mice than you can do to human children, in terms of we're injecting drugs, potentially giving painkillers, things like that. So talk to me about the process around what you can and can't do, and how that ethics is governed. Hitting you first, Jason.
Yeah, a lot of this has shifted over the past, I'd say, decade, where in many cases, and I love animal models because I think they provide so much insight, and I think we'll talk about the insight that they provided. And part of it is because of the precision. There's just that you can control so much more in animal models. Human models, you can't. There have been a couple of inroads into this of late that I find intriguing, but kind of one-off. So one of the things we were talking about earlier when you were asking, you put the electrodes on the head. Part of the problem with so I do primarily EEG work, not MRI work on campus. We don't have an MRI on campus. That means that electrical signals that are produced by areas of the cortex are then bouncing. So if I pick up a signal over the frontal part of the scalp, that does not mean that it's the frontal lobe that's producing it. It just means somewhere in the head produced it. So they're really, really bad at spatial aspects of what's going on. they're really good at timing. I can get down to millisecond precision of when something happened, but milliseconds is still a long time in brain reactivity. So a lot of people have started to take advantage of something called ECOG units, but it is implanted electrodes that happen in the couple of weeks before seizure surgery in pediatric populations. Wow. So oftentimes, couple of weeks before an individual who has pretty severe seizure disorders is going to have surgery, they actually implant electrodes so that they can directly map a lot of one-to-one correspondences of areas of the cortex with specific functions. So language functions, for instance, so that they know exactly where language aspects are so that they hopefully don't interrupt that during the actual surgery that will take place. That's provided an interesting window where several labs have started to ask also, I used to work in one that was starting to do this, but questions about empathy, questions about morality, where you can actually get at the exact locations at the exact time they're doing the processing, not 4 to 6 seconds after they're doing the processing. And so it's a really cool new thing, but you're talking about sample sizes that are 2 or 3 kids over a year. So they're not generalizable findings, but that's the closest to the kind of precision that I think animal research is. Otherwise, I need to be non-invasive. So everything I'm doing is very much about how do we protect, and not just we, but our IRB, which is our institutional review board that's going to go through any kind of things I want to do with kids, with adults, any kind of games that we're going to play. And they're going to look for any potential harms that could come or any risks that especially researchers might not think about. And they're going to ask you to clarify a lot of things and to be really straightforward to the kids and their families or the adults, college students coming into the lab, to know exactly what they're signing up for, what the potential risks are, what the benefits could be from this. And so you're always weighing those out. It honestly falls most of the time to you can't do any kind of actual drug administration in a lot of stuff, although there are places that do drug administration studies. With kids, the only real one that's been out there is there's been a long-standing one of nose squirts of oxytocin that were supposed to help. It's an Australian study from about a decade ago where they tried an intervention in kids that were on the spectrum to see if oxytocin administration would help. That one raised a lot of ethical flags for a long time in the social neuro world, where it was, can they actually consent to doing this? We don't know what the long-term implications of a drug administration like that are on kids.
Yeah. Wow. So that's fascinating. So I thought that in brain neural implanting an electrode in there was a big no-no, but it sounds like, so when it's related to the treatment of a disorder that a child is suffering, then it's, I guess that opens a door, because now we're doing it for something that's beneficial to the child. We're not just doing it to satisfy our research curiosities. That's
exactly it. Yeah. So it's very much just asking research questions for the sake of research, which is a joy in many ways. It can't be at the forefront of how those kinds of studies go. You have to be very aware of the risks. And in those cases, it's also about convenience. It's about making sure that the kid that is about to undergo surgery is not inundated with everybody's different studies going on.
Sure. And you're both parents yourselves, and I'm sure you wouldn't necessarily sign your own kid up to have an electrode implanted for no good reason.
Most definitely, yeah.
Right. All right, Todd. So I think the assumption is in the general public that with animals we can do anything we want, but I think you're going to tell me that that's not true.
Well, we can do a lot more, but we are governed by something called the IACUC, or the Institutional Care and Use Committee for Animals. And so I actually serve as the chair here at UWGB. We have a great committee here that's very understanding. So that committee actually is composed of scientists, also a community member, and a vet. So what we have to do is we actually have to, they do this for human subjects too. It's just done through the Institutional Review Board, or IRB. So we have to submit our studies. We have to explain what we're doing, why we're doing it. We have to explain how many mice we're going to do. If we're using any kind of pain, we have to say if we're doing any pain or stress and how we will alleviate that. So there are a lot of ethical considerations that you have to have in there. You have to explain if you are going to do drug administration, where are you getting your drugs from? How pure are your drugs? There's a lot of safety that goes in. It's not just a free-for-all. It's not the wild, wild west where you just get to do what you'd like. But you can put them into protocols, and most of the time, again, RI Cook's really good about giving feedback or helping you make the best decisions. But in my career, I have done a lot of different things. So I have done a lot of electrode implantations in mice, or actually in rats, to be able to look at neurochemical levels. So in those studies, we were trying to find drugs that helped improve cognition. So we were looking in the prefrontal cortex area, which is also a debate we can get into later on if mice have prefrontal cortices or not. But I've done that. I've done where we've implanted electrodes and they would actually respond on a lever because they found it pleasurable. But in our lab, we don't do any kind of invasive, we don't have the technology for it. So again, what we're doing is a lot of just behavioral observation. We do a lot of drug administration. So a lot of our probing is done through selective agonists, which activate receptors, and selective antagonists. So that's kind of how we probe the brain. So we're not actually, stimulating neurons or anything like that through electrodes, but we can use drugs to try to activate, specific neurons. So if we wanted to activate the glutamate neuron, we can do that, or we can activate the GABA neuron, and we can get very specific. So I think we do have a lot more flexibility in that we don't have the same ethical concerns as humans, right? We're not as concerned about the long-term health. If you go back and you start thinking about some of the early psych studies that were done that we found had long-term implications. So with the mice and their lifespan, we're not worried about those types of issues.
But still important to make sure they're not suffering unneedlessly.
Yep.
Yeah, absolutely. All right, so I do want to get into that debate about whether mice have prefrontal cortex. So I don't know who wants to take this one, but I wonder if you can explain to me, because I haven't studied this since I was an undergrad, the human brain versus the rodent brain. What is the difference? What are the similarities?
So there's a lot of similarities, a lot of differences. But I think one thing to keep in mind is that questions we ask in mice don't always directly translate, and it's not an end-all, be-all, right? We're using it to get insight and ask questions we can't ask in humans, so then maybe we can find a way to ask it in a human. So one way to put this is that I've also worked in molecular labs, which were tough for me. I felt it was kind of boring sitting at a desk and working in a petri dish, but that's my own personal opinion. But a lot of times you start out that, right? We start small and we move up. And so there are a lot of times in drug development where they use cells, right? And then they say, oh, I got this really cool drug, or I got this really cool finding in these primary neurons or this cell. And then they go and put it into an animal or into a human, and it flops, right? Because we don't appreciate in cells, we can get information, but it's also a very closed system. There's not a lot of counteracting systems that are fighting with each other like we have in a whole intact brain. So I think the mice provides a lot of information that we can't get from cells. So it is a step up from cellular studies, but we can use our hypotheses from the cellular studies. But I think we run into the issue of how relevant is what we're doing in mice. And I think my field specifically with the behavioral stuff is one of the bigger questions, right? Mm-hmm. So that's why my lab tries to do more translational models or work on translational models. I'm actually working with Herb Covington, who was here at UWGB on a special issue, discussing the different translations and trying to push forward how we should start considering models, right? And what the actual outcome is and how close it is, because we do see a lot of drugs that work in animals and we're like, yay, we've got the new best antidepressant. And then we put it into humans and it fails. Mm-hmm. And so we're saying the big question right now, I would say, is the animal models we have are, we say they have good predictive validity, right? So an example would be force swim. You place an animal in a container, they swim for a little while, then they stop. And what we measure is called immobility time. And so immobility time is basically them just laying there, right? People in the past have said, I don't agree with this, so I just want to, they said they've given up. It's learned helplessness. They stop swimming because they become helpless, they can't escape. I would argue they've learned that, hey, I can float, right? Because you have to remember that these are mice that have lived in a cage. They've never experienced water most of the time. If we use them if we expose them to the forced swim test multiple times, they actually, each time you do it, they spend more time immobile. They spend more time floating. So to me, that's a learning thing, right? They're like, I don't need to exert energy. I'm going to save my energy. But that's one of the biggest screening models for antidepressants. If you give them something like Prozac, right? Prozac, they'll actually swim a lot more. So the immobility, the quote unquote learned helplessness goes away and they swim a lot more. And so a lot of antidepressants work in that task. So we think it has really nice predictive validity. However, when we test new drugs, they don't work in it. So then we say, oh, this drug's not going to work in a human. But the question becomes is the forced swim testing like serotonin? You know, so if we're looking at a new mechanism, is it not showing because it's a totally different mechanism and it's not exerting that behavior? So there's a lot of questions on, our animal models really focused more on drug activity as opposed to the behavioral outcome or the translation we want. And so there's a big push for better models. They're starting to come out with models where instead of food being the reinforcer, like there's social reinforcers, there's sexual reinforcers, things that us humans like, right? You know, we like to eat, but we also like access to drugs or access to friends or access to sex and things like that. So the field is really moving and starting to consider what is more relevant or what does it actually look like in a human? And so we have to also appreciate that some of these models will fail. And hopefully people will publish negative findings so that we don't all just keep trying the same thing over and over again. So
Right. Yeah, because it sounds like if it's possible with the swim test that you're talking about, that a potential drug that may actually be useful in humans might get sort of cut off prematurely.
Absolutely.
And that would be a real shame.
Absolutely.
Yeah.
Yep.
Jason, do you have anything you want to add about what's special about a human brain?
Well, not necessarily what's special about well, I guess what's special about a human brain in some ways, but I think even if we're talking about neuro outside just the petri dish, so to speak, I mean, there are models in fruit flies. There are models that, like, you work your way up. Some of the coolest findings, I'm thinking the memory finding of the new synapse formation. I think it's in a sea slug, one of the original ones, to say, like, there's an actual new synapse that represents a new fear-based memory. And it's fascinating. Whether it replicates to more advanced species is a different question, but I think there's so much insight that's coming forward. A lot of the more basic functions, I think, you can replicate really well in animals, especially if you're talking about the majority of definitely mammals are going to have some of the brain stem-ish sort of features, some of the other elements right above them. When you started, you were talking about prefrontal cortex. And so I think that's one of the bigger debates is like the extent to which some of the more CEO-based aspects of the brain that could be unique in humans, although that's questionable because I would say many of the great apes have a variant that's comparable. Sheep may have a variant that has some relative PFC aspects. I think there's a world where rats and mice have variants of this as well. So the uniqueness to humans isn't necessarily there. And even when we get to things like language that we would consider remarkably unique, if you get into neuroscience with songbirds, you get something that really closely looks like language in areas that really closely look like a Broca's-ish, a Wernicke's-ish with a arcuate fasciculus connecting them. So different areas connecting them, but you get kind of a languagey circuit that is active, that if you lesion, interrupts song production in this. So it does seem like many of the things we thought were unique to humans are represented differently. And so I think that's the challenge, but analogously, I'd say.
Okay, so it's not that so when we're choosing an animal to do research on, you mentioned there's a whole variety that people do. You want to pick the species that it closely mimics that particular skill set. So a songbird might help you study language in humans, but a mouse might help you study more drug use in humans. So how do people learn which animals are best for what?
I would say based on publication, communicating with other scholars, looking up the data. I would say that's kind of the driving force. To me, I think it's also, I think sometimes we get away from this. For me, I've always been very cautious when I report my findings. I do publish, but when I do it, I try to always make sure I'm publishing within my context. I try to always make sure I say, this isn't an animal. This is an antidepressant-like behavior, an antidepressant-like effect, because that mouse didn't tell me they're depressed. So I don't know if they're depressed or not, no matter what assay we're doing, right? So there's somewhere, if it's a more abstract concept like depression, I try to kind of couch my findings into this is what I found in this mouse, right? Like I try, there are other researchers that will say they cured cancer, right? But I try to, I think if we can make sure we keep that in the realm and keep it in the context and not overgeneralize, I think there's value we can learn from all of the, different species and different types of science.
The way we've been talking about this so far, if I'm getting it right, is that we're building these animal models, possibly with, by the sounds of it, smaller animals first, and getting to maybe primates and bigger animals, and then generalizing to humans. Does it ever go the other way? Do we ever take human research and apply it to animals?
I'm sure we do, but I don't know that many models.
I think we would. It's like more of the models, right? Like we're trying, or it's more of the disease states is what we take, and then we try to find a model. And so that's kind of what we're taking from them, or we try to look at, if you see that this disorder has, like in schizophrenia, there's a lot of cognitive disorders, a lot of issues with working memory and executive function. So you can take that and try to build a model that also, or deplete certain neurons or receptors in the brain to produce a model that maybe has that same issue. So that's kind of almost how we use humans is like, okay, how can we make a disease state that looks like that in an animal so that we can solve that problem?
And even, that's a good way of saying it, actually. And it's got me thinking, too. If you look at some of the original, I'd say one of the aspects that can sometimes happen in animals, although it's ethically, is lesion studies. So lesion studies you can't do in humans. Well, ethically. Shouldn't do in humans. Yeah, we'll go with that. But for instance, some of the early models of where is memory stored in the human brain were, there must be a specific place. And then I'd say the best information was taking that model of it should be everywhere. And then I think it was Lapsley that started removing parts of a mouse or rat brain. Yep. And it ended up being, it's like 60 or 70% of the cortex had to be removed before.
Cut it all
he just had to keep lesioning before the mouse or rat actually lost the engram or the memory that was there. And so I think that's a really good model of going, hey, we think one area of the brain, let's lesion that. That didn't actually work, and then going to whole scale.
So if you have a hypothesis about the human brain, you find a way to test it in the animal.
It is more testable in many ways, yeah.
I would even say, even going back to the point of like, what have we kind of taken from humans? A lot of drug development actually happens serendipitously. So a lot of our first antidepressants and the drugs for schizophrenia, the antipsychotics, those were not developed for that disorder. They were developed for like tuberculosis and things like that. And this is back before we had the IRB, like we talked about before, where chemists could send their drugs to their friends who were doctors at like psych wards and then administer them to the patients. And so that's actually one of the biggest reportings was when people reported they were taking imipramine and they reported a feeling of being lighter and not having depression anymore. And then they went, wow, this is an antidepressant drug. How does it work? So they actually gave them the drug, didn't know how the drug worked, it produced an antidepressant effect, and then they had to go back into animals and into the cell studies to find out how this drug works. And then that's what drives that's what's been driving our hypotheses and what's been driving some of the drug development.
Yeah, no, and I am no drug expert, that's you, but there are quite a few drugs that we use today that we're not entirely sure how they work, right? And so we're still trying to figure that out in animal models?
Yep.
I mean, the most common SSRIs. Yep. Still unclear how it exactly works.
Yeah, there's a lot of different hypotheses on that. Ketamine is another one. Ketamine broke out, I think it was, probably almost 10 years ago, more than that now. It's become more prominent, but we still don't know how it works because we've developed drugs that look just like ketamine from a receptor standpoint of where they activate receptors or inactivate receptors and things like that, and they don't work like ketamine. So there's something unique about ketamine, and we're not quite sure. There's a lot of hypotheses out there. We published data from the lab here at GB saying that ketamine's effects require opioid receptors. Other people have found something similar. There's a lot of things where we just, sometimes they work in humans, and then we have to go back and find out what's unique about that drug to kind of help us move forward.
Yeah.
And we may not figure it out. Sometimes I think it might be the combination of that drug hitting multiple receptors that produce the effect, not just a one receptor site.
Yeah. Yeah, and you've talked a lot, Todd, about, publishing and sharing your research with other areas. And I think that that's what I'm getting from you both, is that no single lab is going to come up with the answers, right? It's a combination of multiple labs and, I guess, the great scientific process.
Yep.
All the work.
It's rarely. There are some mass labs at some of the larger R1s that go all the way from cell through applied human work, but those have 15 postdocs, 30 grad. They're just massive labs in neuro. The majority is coming from specialists in different places that are then trying to build models together.
Yeah. So if any of our students listening to this want to be involved in either of your labs, do you mind giving a little pitch on how they can get in contact with you if they're interested?
Yeah, email me or stop by my office. I'd love to talk to you about this. It is, I think, one of the privileges of being where we are is getting to teach these things that we love doing to people like EEG, that you can actually do the hands-on pieces. So come talk to me. Love to get you into it.
Yeah, same goes. I think email works, so you can stop by my office. I'm trying to get a little more into the human research, even though we talked about some of the complications, but just to open up and have more options. So I'll probably be doing animal work and human work. Yeah.
Fantastic. Well, thank you again both so much for coming on the show and talk to me about animal and human brains. Really appreciate it.
Always fun. Thank you.
Thank you. Psychology and Stuff is brought to you by Good Cause Productions at the University of Wisconsin, Green Bay. The executive producer is Ryan Martin, the production manager is Rachel Sankey, and our creative director is Jenny Young. The media engineer for this episode is Kennedy Punky, supported by Rachel Sankey. Special thanks to my guests today, Dr. Jason Cowell and Dr. Todd Hillhouse. If you haven't already, please make sure to rate, review, and subscribe to us on your favorite podcast platform. I'm your host, Allison Jane Martingano. Keep being amazing.